Okay, hello everyone. We are now live, and thank you for joining us to this ISCB-SC webinars sponsored by International Society for Computational Biology. So I would like to remind you, if you want to know more about the upcoming webinars or how you can present your research and be part of this initiative, you can follow us on Twitter as @ISCB-SC. So today we have an interesting talk by Juan Sebastián Andrade from Universidad de los Andes, and his presentation is titled "Exploring the Herpesvirales, Core Genome and Evolutionary Relationships." So thank you, Juan Sebastián, for joining us today here, and please, the floor is yours.
Thanks a lot, Jesse. Okay, hello everyone. Thanks a lot for being here. I'm going to talk today a little bit about a project that I've been working with for the last 4 years, which is called, as Jesse mentioned, Exploring the Herpesvirales, Core Genome and Evolutionary Relationships. So, as I mentioned, I began this project about 4 years ago, and one of the first texts that I read about viral evolution, since we're going to be talking specifically about that, was this book chapter by Roger Hendricks, which gives us an idea of how hard it is to work with precisely the evolution of viruses. And he begins the chapter by giving us some figures to demonstrate this point. So as you know, viruses are 5 to 10 times more abundant than prokaryotes in a given environment. They are the most abundant biological elements on Earth. It is estimated that there are about 10 to the 31st power viral particles in the biosphere. And when you take a look at marine ecosystems, and especially at phages in marine ecosystem, it is calculated that the total biomass of viral particles is renovated or has a turnover that takes place each 4 to 10 days. So that leads to estimates that there are about 10 to the 24th power productive viral infections per second. Productive infection being one that leads to the creation of new viruses. And at that point, I stopped reading and looked at this astonishing figure and thought, well, 10 to the 24 is a lot. Just to give you an idea, that's more than the total number of cells in your body, than the number of people that have ever lived, than the age of the universe in seconds. It is comparable to the number of stars in the observable universe, assuming that there are any exoplanets with viruses, which I firmly believe they are. But nonetheless, it is more than a mole. And as you know, a mole is a lot of particles. And each of these productive infections, since it leads to the creation of new viruses, represents an opportunity for the occurrence of an evolutionary relevant event. And this is going to be really important in a second, so hold that thought for a while. I'm going to stop this part of the discussion to present the main protagonists of this particular story, which are going to be first the order Herpesvirales. These are enveloped dsDNA viruses, that is double-stranded DNA viruses. The majority of them have vertebrate hosts, although the smallest family does have bivalve hosts, so invertebrates. And they are really interesting in public health because they are known to produce chronic infections in quote-unquote healthy individuals. So depending on specific herpes virus you're looking into, you can find a prevalence of about 90% of the population. So whether we like it or not, most of us have herpes. When you're working with viruses, and in general with any other clade, be it from viruses, prokaryotes, eukaryotes, whatever, you can define a concept which is called a core genome. A core genome is that set of genes and by extension proteins that were harbored by the common ancestor of all of the members of the clade you're talking about. In the case of Herpesvirales, you don't have a core genome, but we do have a core genome for the biggest Herpesvirales family, which is called the Herpesviridae, which is composed of 43 genes and was defined in 2006. And with that being said, the second main protagonists are going to be the Caudovirales, which are the theophages, which is a humongously big order of dsDNA phages, which have prokaryote hosts, both archaea and bacteria. So the reason why you'll see that these two orders are frequently talked about conjointly is because we have seen over the past 30 to 40 years that they have similarities in their viral cycles, that they have common faults in certain structural proteins, and that there's some conservation in the ATP subunit of the DNA terminus, a protein related to the synthesis of nucleotides. So this has all led to believe, again, for the past 4 years or so that there is a common ancestor between these two groups. Thing is that, again, we're working with things that mutate a lot due to the high number of productive viral infections per second, especially in phages. So if you try to do an alignment of certain Herpesvirales and certain Caudovirales sequences for this DNA terminus ATPase subunit, you'll see that there are the number of regions that do have certain degree of conservation, which is represented in this image by asterisks in the rectangular orange box, is kind of small. And these are just portions of the subunit, not the complete sequence. And again, this all leads to the fact that when you're working with viral evolution, you're working with elements that have relatively small genomes, at least compared to most cellular organisms, and have really high mutation rates. So whenever you're working with viral evolution, you have to surpass a wall that's represented by this problem. In my lab, we use two approaches to try to deal with this. So first one is called viral domain orthologous groups, or VDOGs, which I'm going to talk mostly about in this presentation. And the second one are K-mer frequencies in complete genomes, which I'll talk a little bit about towards the end. So the objectives that, for this specific part of my 4-year-long odyssey were first to elucidate the evolutionary relationship between these two orders. Naturally, that was the big objective. More humbly, what we wanted to do was just to provide evidence of common ancestry through our approach. And we saw that it was possible as well, simultaneously, to define a core genome for the Herpesvirales. A core genome, again, being that set of proteins and by extension genes that were harbored by the ancestor of the complete order. So all of these, all of the analysis I'm going to show you are based on a master's degree thesis from my lab from 2016 by Leonardo Moreno. What he did was basically he defined these VDOGs, and I'm going to explain the next slide more specifically where VDOGs are. Suffice it to say for now that they are orthologous groups of viral protein domains. So this master's degree thesis by Leonardo Moreno defined those VDOGs based on the information of complete viral genomes that he could find in NCBI in early 2016. And what he did was, after defining those VDOGs, he generated an HMM for each of the VDOGs, and he generated then an HMM database for the complete VDOG dataset. So we already had that. We also had the replicated set of Caudovirales proteomes to work with. So the first thing that I did was, being that we already have this VDOG database and the replicated Caudovirales proteomes, was that I did a replication of Herpesvirales proteomes. So what I did more specifically is that I downloaded from NCBI all the Herpesvirales complete genomes that I could find in late 2016. I did a replication at the genome level and then at the protein level. And I ended up with a series of representative Herpesvirales proteomes and associated representative Herpesvirales genomes. And having this, then what I did to check which VDOGs were harbored by each of these proteomes and by extension genomes, I just took all of the proteins from these representative proteomes and did a hammer search against the VDOG database. And this yielded the information of which VDOGs were present basically in my whole dataset. And with that being said, let's delve now more specifically in what a VDOG is. So a VDOG is a cluster of orthologous viral protein domains. So we're working not strictly with proteins, but with domains within the proteins. When you do the kind of search that I did, in which you have a set of proteomes and you do hammer search against this VDOG database, then you can distinguish 3 types of VDOGs. The ones that were detected at least once in your proteomes, the ones that are abundant, that is, they are present in many of the proteomes, and the ones that are representative for the specific clade that I'm working with, in this case, Herpesvirales. So the ones that matter the most for this part of the analysis are those that are representative, because those are the taxonomic terms of the order in this case. That is, those are the protein domains that are unique and specific for that given viral clade as opposed to other viral clades. So how do we determine those representative VDOGs? So there are 3 criteria based on what I just mentioned. So first one is that they should be specific and precise to classify a given group. They should be unique for that given group, which is something that you can derive from mutual information. And if they are unique and specific and precise for that group, then if you plug those VDOGs as features in a random classifier aimed at determining which proteomes belong to that specific clade or not, then they should yield a good result as well. So we determine which VDOGs satisfy this criteria. We label those as the representative VDOGs of the order Herpesvirales. And then, using the information of presence, absence of these VDOGs and neighbor joining, we created a dendrogram, both of the Herpesvirales and Caudovirales, using these representative Herpesvirales VDOGs. And we got something like this. So in this case, we use 2 viral orders and 1 viral family as outgroups. They have either RNA or dsDNA genomes. I'm referring to the outgroups. So what you see here is basically that Herpesvirales and Caudovirales are members of the same big branch, which is distinct from the outgroup branch, which suggests an evolutionary relationship between the two. But perhaps the most interesting thing here is that if you take a look at the Herpesvirales branch, which is the green one, basically of the green that you see here, all of Herpesvirales is in a single branch. So we can say that it is monophyletic. If you take a look at the families of Herpesvirales, which are Herpesviridae, Alloherpesviridae, and Malacoherpesviridae, you see that they are also monophyletic. They are also clustered in the same branches. If you take a look at the subfamilies within Herpesviridae, they are also monophyletic as well. So everything works as intended. However, if you look at the Caudovirales, you see that there are 2 Caudovirales branches, and there's a really big one and a really small one. And the thing is that while the subfamilies that were defined at that point in 2016, 2017, were mostly clustered in the same branch, so they were monophyletic, this was not the case for the families. Again, the families that were defined at that point, because the classification has changed. If you take a look, for example, at the dark orange, which represents the family Siphoviridae, you'll see that there are orange branches all along this big Caudovirales branch. So this is due to the fact that the 3 traditional Caudovirales families, so their classification has been put into question recently. And basically, what we ended up doing, as I mentioned towards the end of the presentation, was that we then moved on after we worked with the Herpesvirales to try to redefine these Caudovirales families. But for now, suffice it to say that Herpesvirales and Caudovirales are part of the same branch, which suggests an evolutionary relationship between the two. So next, we move on to the second objective, was the core genome. So when looking at the core genome, again, as that set of proteins and by extension genes that comes from a common ancestor, we were looking not only at representative VDOGs, but at abundant VDOGs. We wanted those VDOGs that were both abundant and representative due to the definition of the core genome. So we define abundant VDOGs as those which are present in at least 60% of the members of each of the Herpesvirales families. And we did a heat map of those VDOGs that were both representative and abundant. And we got a total of 8 VDOGs, which were associated to 4 Herpesvirales proteins: the TPS subunit of the DNA terminus, both subunits of the ribonucleotide reductase, the DNA polymerase, and the DUTP diphosphatase. All of these proteins are related with DNA synthesis. So in this heat map, what you can see is that, in general, those VDOGs are relatively abundant. There are 2 VDOGs in particular for which you can see that there is a gap for some Herpesviridae genomes. That's due to the fact that for one of the subfamilies of Herpesviridae, which is called Beta-Herpesvirine, some of the domains associated to these VDOGs are either inactive or absent, and this is something that has been seen in the literature. So those gaps make sense. But the most interesting thing here is that if you take a look at the distribution of those VDOGs in Caudovirales, you'll see that most of them have the DNA terminus, which was expected. But for the other VDOGs related to the other proteins, you'll see that their abundance in Myoviridae is relatively higher for some cases as compared to their abundance in the other 2 families of Caudovirales. That's going to be important in a second. So what we did after this was that, having detected these abundant VDOGs, we took the most abundant VDOG in the Herpesvirales for each of these proteins, and we tried to generate a phylogenetic tree using basically the sequence information. Because if the core genome comes, if the sequences of the core genome come from the common ancestor of the whole order, then if you use them for a phylogenetic tree, they should reflect the overall history of the order as a whole. So we did an evolutionary tree with Raxemel, a maximum likelihood tree. And we got something like this, in which we use Myoviridae phages as outgroups. So what you can see is that the bootstrap values for the main divisions of families and subfamilies are relatively good. In general, the bootstrap values were reasonable even for the smallest branches. So there's a consistency in the evolutionary history that is told by these proteins as compared to what we see in the literature. There are certain differences in the details of the dispositions of the subfamilies, but you have to take into account that prior evolutionary trees of the whole order Herpesvirales were mostly done just with one sequence, and here we're using 4. So it makes sense that our the information given by our tree is much more robust in those details. The last thing that we wanted to do is that there is some evidence that the core genes of the family Herpesviridae, which, as I mentioned before, were previously defined, had some degree of synteny. So we wanted to see if the genes that are associated to these potential core genome proteins had as well some synteny, in the sense that they were clustered together in the Herpesvirales genomes. So what we did was that at the family level, we generated null distributions of genome distances between the genes. And we compared that null distribution generated through randomization to the observed distribution of gene dispositions in the real Herpesvirales data. We compare basically the median and the distribution of these two distributions, the randomized one and the real one. And what we found was that for the Herpesviridae and up to some point from the family Herpesviridae as well, there is some degree of synteny, or some evidence of synteny, of clusterization of the genes associated to this core genome. For the family Malacoherpesviridae, we got inconclusive results because the number of proteins that we're working with was too small. But the interesting thing is that if you take a look at the family Myodiridae from Caudovirales, which I mentioned before, there's also some evidence of synteny, at least on phages, that is of clusterization of these genes associated to the core genome. So taking all of this into account, all of the results that I mentioned, what can we conclude? So first thing is that the dendrogram does suggest that the Herpesvirales and Caudovirales are sister clades. However, the analysis that we did regarding the core genome yielded some interesting results, which were that many Myodiridae proteomes harbor the abundant Herpesvirales VDOGs, as we saw in the Heatmap. Myodiridae is actually the only family where we could find outgroups for the phylogenetic tree. And Myodiridae is the only Caudovirales family with some evidence of synteny of these potential Herpesvirales core genomes. So what we think that happens is that either the Herpesvirales and Caudovirales are sister clades, or that Herpesvirales comes from Myodiridae, or one of the subfamilies of Myodiridae, most probably T. virine. And naturally, the core genome proposed for the Herpesvirales is composed of the 4 proteins that I have mentioned. The ATPase subunit of the DNA terminus, which was the one that we knew prior to the analysis that had to be there, the ribonucleotide reductase, the DUTP phosphatase, and the DNA polymerase. So that what I mentioned and what I wanted to speak about was about half of the work we're currently doing. So that was like the first stage of the project. What we're working in right now, and we've been working for the last year and a half, 2 years or so, is to move forward and to take a look at not at the Herpesvirales, but at the Caudovirales. And it's important to look at the Caudovirales because they are the most abundant dsDNA of each order, and they are really ancient. So we're not only looking at the Caudovirales and their classification, which has also changed a lot due to recent ICTB taxonomy releases, but also any potential relationship that they can have with Detectiviridae, which is a small phage dsDNA phage family, which gave rise to what we call the polynthus, which is a series of selfish genetic elements that infect mostly eukaryotes. And what we want to do here is not only use VDOGs, but also take a look at how K-mer frequencies can help and complement VDOGs to generate an approach to viral classification. So the results are still inconclusive. We're still working on that. Suffice it to say right now that the most encouraging information that we have is that if you use both K-mers and VDOGs for generating a measurement of distance or relatedness between viral genomes, you see that there are some Caudovirales phages which are more close under this measure of distance to Detectiviridae phages than to other Caudovirales phages, which is a really indirect sign of a potential relationship, although this is not conclusive as of now. So we're still working on that. We're working on measures to define families, subfamilies, and order based on K-mers and VDOGs. Then we can revalidate the classification, and perhaps above that, check if our method of classification is consistent with what the ICTB has done in the last couple of years. And naturally, since the VDOGs are viral protein domains, we're trying to annotate as best as we can these viral proteins domain just to have information on the specific functions that they are associated with. And with that, that's what I wanted to share. So again, thanks a lot for being here. Special thanks to all of the people in my lab, to Jesse and the Complete Student Council for allowing me to share this part of my research. and to the high performance computing cluster at Universidad de los Andes for allowing me to do all of the computational work that I had to do to work in this and continue to work in this. And with that, I'll open myself to any questions.
Okay, thank you, Juan Sebastián. Can you hear me?
Yeah.
Okay, so it's time for questions. Let's wait because we have a delay with the YouTube broadcast. But in the meantime, I have a question, or maybe if you want to talk about what are the challenges in terms to reconstruct the evolutionary relationships in viruses, or maybe in your case in Herpesvirales. I mean, in terms of the different exogenous sequences or genomes that are integrated in their genomes. To be more specific, what are the noise that can introduce the phages in the genomes in order to reconstruct the evolutionary relationships?
Okay, so that's a really good question. So the thing with phages naturally is that you're dealing with so there's this specific term which was coined by Ackerman, which is a really important a really important researcher in Caudovirales in particular. In fact, one of the new families of Caudovirales is called Ackerman in his honor, and he stated that any phage genome has some degree of mosaicism, which means that we usually see a gene, at least for cellular organisms, as a set of proteins that evolve over time. This particular true for eukaryotes. Naturally, for prokaryotes, you have certain degree of horizontal gene transfer. But one conceives usually a genome as, again, mostly a set of genes and transcription factor, and all of these regulation elements that evolve through mostly through common ancestry and through mutation over that common ancestry. For the case of phages, you have a mosaic in which certain genes come from certain events of gain or loss of genes, and certain genes might come from a common ancestry or not. But so the main challenge is that we, up to this point, don't have a 100% accurate way to determine which genes come from where. So if you want to, for example, define what I'm trying to do in Herpesvirales, a core genome, that's going to be a little more difficult, because you can't say that, okay, gene A comes from a common ancestry of, for example, the Caudovirales for detectivity, and gene B comes from horizontal gene transfer, or an event of gene transfer between viruses, not between the phage and its host. Like that. So that's the main noise that we have in phages. And the other, naturally, is going to be the fact that, so this is our approach to viral classification, but there are many other approaches possible. So depending on what you want to work with. So I think that the most important source of noise right now, and this is noise in a good way because it's final research, is the fact that we have to sit down and try to come up with measures to define viral classification above the genus level. This is something that the ICTP has been working a lot in the past, basically since its creation, but mostly in the past 2 to 3 years. But until we have a clear-cut and universally possible criteria for viral classification, we'll get not only noise based on the ways that viruses evolve, but also in the ways that we're trying to approach it. So luckily, we can solve that problem in the next 5 to 10 years.
Thank you, Sebastián. I think in the same way, it is usual to find in the core genome of Herpesvirales, or if you look in other virus, to find those phages genes that are shaded in common with different virus, or the phages genes or the phages DNA is usual to find in the specific genome of every virus?
Okay, so also a really good question. So we have to take into account that I'm afraid it really depends on the specific case. So something really interesting about viruses is that we can't ascertain at this point that So let's take a look at cellular organisms. So we do have a tree of life for cellular organisms, right? So we can ascertain from that we all have a common ancestor. We share at some point of the evolutionary history of Earth, we shared a common ancestor with E. coli or with Staphylococcus aureus, or with bacteria and archaea. For viruses, we really don't know. So some viruses might come from have their specific lineage of evolutionary history distinct from other viruses. So what we know is that in general, for those eukaryote viruses that come from phages, there should be at least some genes that were retained in those eukaryote viruses due to common ancestry, which is the case in Herpesvirales. But being viruses self-genetic elements that are specialized to attack certain cells and by extension certain organisms, evolutionarily speaking, for the virus, it is more important to have those genes that are required to adapt their process of replication to their specific host. So what you see in most cases is that the second team primes over the first. So in general, what you're going to see is that most of the genes, at least the core genes, the ones that you find in a core genome for a given viral order, are mostly shaped by the host that they co-evolve with, rather than by their evolutionary history. And this is true in the case of the Herpesvirales as well. So all of the genes that make part of the Herpesvirales core genome that we proposed, they are related to DNA synthesis, but they are shared between eukaryotes and prokaryotes. So basically, to give a particular example, the ribonucleotide reductase is a protein that is present in us, is present in our eukaryotes, is present in our bacteria, and present in our archaea. So it makes sense that in the transition of the Caudovirales from being phages to what we have as the Herpesvirales, they would retain those specific proteins. So they come from the phage, but they were retained due to the fact that we as eukaryotes also use those proteins. So it makes sense for the Herpesvirales to keep those proteins in particular. So there's really an interplay between those two things. But I say overall that the host is really the main factor that mediates a core genome, not really the evolutionary origin.
Okay, thank you. Yeah, it is because also the phages respond more to, maybe to adaptative, to adaptative process depending of the environment, right?
Yeah, that's completely right.
Yeah, that's what happens with bacteria and the CRISPR arrays. You have different kind of CRISPR arrays depending of the phages that are surrounding the environment. Okay. So thank you, Juan Sebastian. So I think that we have no questions from attendees. So once again, thank you, Juan Sebastian.
Thanks a lot for having me.
We hope to have you here in our webinars in the next opportunity, and take care. Thank you.
Okay, thanks a lot. Bye-bye.